I'd say that also this time will be in English if the speaker agrees.
Okay.
So in this second English session of the theoretical biology seminar, we have Svetlana Treta from the neurophysiology department of the National Institute of Psychiatry here in Mexico City. So who will talk about kind of new type of phenomena, or new, well, at least a model type of synaptic plasticity in, how do you say Sangujela?
Leech.
In the leech. Ah, it's leech. Thanks.
Thank you very much to Alessio and Narco for the invitation. Today I'm going to talk about work done basically by Michelle Sanchez in my lab. And so let's begin by some background. At chemical synapses, neurotransmitters are packed into synaptic vesicles, which are these bags of membrane containing molecules of neurotransmitter. And in order to release this transmitter and transmit a signal to the postsynaptic cell, these vesicles need to be fused with the presynaptic membrane here. And for that, when electrical activity occurs and an action potential like this invades the nerve terminal, calcium channels, which are activated by voltage, are opened, and those calcium flows following its electrochemical gradient. An increase of calcium occurs in the nerve terminal, and this triggers a series of molecular events that lead to vesicle fusion with the presynaptic membrane. So this bag kind of opens to the outside, and thus releases the transmitter to this space between the presynaptic and the postsynaptic membranes, which is called the synaptic cleft. This is a very small space, so the transmitter molecules diffuse very rapidly to reach the postsynaptic membrane and bind to molecules in this membrane, which are proteins called receptors, which are specific to each neurotransmitter. And these receptors can be either an ionic channel, which directly open when the transmitter binds to them, or can activate a series of intracellular signaling cascades to finally open ionic channels in the membrane. So the final result is that ionic channels open in the membrane, in the postsynaptic membrane, and ions flow in or out depending on what concentrations and electrochemical gradients are. And we get a change in the membrane potential, which I schematically represented here as a depolarizing response. And this is called a synaptic potential. Let me just say, if there's anything that is not clear or you want me to stop and explain, don't hesitate to stop me. Okay. So we have a presynaptic action potential and a postsynaptic synaptic potential. And all this was studied with a lot of detail in the 50s by Bernard Katz and his groups. And Del Castillo and Katz studied how the neurotransmitter release was regulated, and they basically found out, even before we knew about electron microscopy and we could see vesicles, they found out that transmitter release occurred in packages of of a constant content, which they called quanta. So they proposed the quantal hypothesis for synaptic transmission, which basically postulates that the number of quanta that are released by the presynaptic membrane depends on the number of quanta that are available in the presynaptic terminal and the probability of release. And we will see that this probability is basically changed by the calcium concentration in the terminal, as I already explained. And this N means basically the number of vesicles that are available in the releasable pool of vesicles. Okay. So the mean number of quanta that are released varies when there is repetitive electrical activity, because there are plasticity phenomena, and there are plasticity phenomena that occur in a short time, which are called short-term plasticity mechanisms, and there are also some longer processes which I won't talk about today. But short-term plasticity phenomena, the most known are synaptic facilitation, whereby the second of two presynaptic action potentials produces an increased synaptic potential because more neurotransmitter is released with this impulse than with the first one. And this was studied by Katz and Miledi, and they showed that this is basically due to a residual calcium that stays in the terminal. When an action potential occurs, calcium enters, so there's a very fast increase in the calcium concentration in the terminal, and this calcium decreases exponentially with some time constant. But if a second action potential arrives before this calcium has come to the basal level, then this action potential produces more calcium entry, which sums to the calcium that was already in the terminal. And since the number of vesicles that are fused depends non-linearly on the calcium concentration, that produces an increase in transmitter release. So this is called synaptic facilitation. And now there's another short-term plasticity phenomenon whereby when a lot of transmitter is released with a first presynaptic action potential, then fewer vesicles remain available in the terminal, and that produces a decrease in the synaptic potentials that come subsequently. This happens mainly in response to trains of action potentials, which produce a lot of release, or under experimental conditions where one increases the probability of release by increasing extracellular calcium. So this is basically understood as a phenomenon caused by vesicular depletion in the terminal, right?
Am I wrong if I associate, let's say standard condition, I associate the first one to spikes, to single spikes, and the second more to burst? Or is too simple a picture?
Yeah, both. It depends more on the initial release amount. If the initial release is high, then it's more likely that the vesicles available will be depleted, and then subsequent activity will find less vesicles and that causes depression. So it depends. You can get this with pairs of pulses with high probability of release,
I see.
or you can get it with bursts with low or normal probability, right? And as I said, facilitation decreases with a time course of tens to hundreds of milliseconds, and here depression recovers with a time course of 10 seconds. And these two phenomena can interact in the same synapse.
So depression is lower.
Sorry?
Depression is lower phenomenon.
Right. Right. So these are classical recordings from the nerve muscle junction, and here we see that under low release probability, where calcium was decreased, then we get a first synaptic potential with an amplitude, but the second and the subsequent ones are much bigger than the first one. And even after some time, the synapse still is facilitated. So this is an example of pure facilitation, right? If the probability of release is increased by increasing extracellular calcium, then basically we see depression. Each synaptic potential is smaller than the previous one. And in this case, with normal calcium concentration, we have facilitation in the second pulse, but from the second to the third or the fourth, there's no more facilitation, not like here. Oh, I think my pointer is out of battery. Sorry.
There is one in the.
You have one?
In the.
Okay.
In the console.
Here?
There should be a small.
Laser. Yay! Thank you. So here we have facilitation in the second pulse, but not afterwards. And that's because depression is mixed here with facilitation. So even though there is a higher release probability, there is a lower number of vesicles available, and so we can't increase the number of vesicles fused anymore. And after some time, the synapse remains still depressed, right? So these two phenomena can interact with each other, but there are some preparations, some synapses, including the neuromuscular junction, where there is synaptic depression with two pairs of pulses separated by a long interval. In spite that two pulses with a short interval between them produce synaptic facilitation. So that's not explained, that cannot be explained by vesicle depletion, because basically if you can't get facilitation, there are enough vesicles after one action potential to equal or even increase release. And so why do we get depression with longer intervals, right? And that's the problem I'm going to talk about in this work. And to study that, we used the central nervous system of the leech.
Just a question. So there would be ways to decrease the response in the postsynaptic neuron by taking away receptors, by doing other things, right?
Right.
Adaptation and so on.
Right.
So are you focusing only on presynaptic mechanisms of facilitation and depression for the rest of the talk? Or?
Yes.
Okay.
So let me explain to you why we use this nervous system. The leeches are segmented animals, like the earthworms. And their central nervous system consists in a chain of ganglia that runs along the ventral side of the animal. And each one of these ganglia, well, let me say, there are 21 segmental ganglia, in addition to a cephalic ganglion and a caudal ganglion. And each one of these segmental ganglia controls in a relatively independent manner one segment of the animal. All of these ganglia are similar between them, and they contain only about 400 neurons. Here we see a view of one of these ganglia taken by John Nichols and Dennis Baylor in the 60s. And we can see several things here. So these ganglia control stereotype behaviors, and something for what we would need thousands of neurons, they do with only a few of them. And they do pretty well because, you know, these animals have survived for millions of years. I'm not sure we will make it. And so we can see that these neurons are very big. Most of them are, some of them are about 10 times bigger than ours. And that makes it easy to do electrical recordings. Moreover, they have a stereotyped position in the ganglion, which is repeated from ganglion to ganglion, from one animal to another, and even between species. And that allows to identify the neurons. So most of these neurons have been identified, and we know their function in the animal. We know, for example, which ones are sensory, which ones are motor, which ones are modulatory neurons. We know the transmitters they release, to which neurons they connect, etc.
A question. The segmentation of the leech is defined only on the repetition of the ganglia, or there is another anatomical system of organs that are repeated also?
There are sensory organs that are repeated in the segments. But not everything is repeated. The reproductive system is not repeated. The heart, the circulatory system is just one for the whole animal. So yes, there are other things that are repeated, but not everything. So in these animals, we can study from behavior to single neurons and vice versa. And whole circuits are known, for example, for swimming, for crawling, for feeding, right? So I hope I have convinced you that it's a really nice system to work on. And so in one of these ganglia, we can identify cells and stick electrodes into them. And for this work I'm going to present, we recorded a synapse between pressure sensory neurons called P cells, which there are 4 in each ganglion here. And the postsynaptic neuron in this synapse is this neuron here, which is paired. And this neuron, although we don't know its function in the animal, its electrical activity is pretty well known. We know that there are no active channels in the soma, so action potentials are conducted passively to the soma, and thus when we record in the soma, their action potentials look like a pagoda roof, and that's why they are called anterior pagoda AP, because they are located in the anterior part of the ganglion. So we recorded with intracellular sharp electrodes in one pressure sensory neuron, which is the presynaptic, and one contralateral AP neuron, which is the postsynaptic. And we produced pairs of pulses with a variable interval and measured the amplitudes of the resulting postsynaptic responses. So, yeah.
The variable interval, how did you generate it?
So we inject current pulses into the presynaptic cell.
And each pulse is
we adjust the amplitude of the pulse. These are squared pulses, but we adjust the amplitude so each produces one action potential, and we just change the interval between the two pulses we give. Yeah, I'm sorry I didn't draw the current pulses here.
Do you need to keep the neuron hyperpolarized to avoid that it is kind of
We did that with mainly with the postsynaptic neuron to avoid firing.
Okay.
Because we wanted just passive responses.
So you're on voltage clamp on post?
We tried, no, not voltage clamp.
No.
We tried to do current clamp, but you know, when you have a whole neuron with its branches, it's not easy to clamp. So, yeah, we did some recordings with current clamp, but it's basically easier to bridge balance and control the current you inject to keep a constant membrane potential. Yeah. Some experiments were done with current clamp. Okay. So we measured the amplitudes of both synaptic potentials and calculated a plasticity index by dividing the amplitude of the second by the amplitude of the first one. And if like in this case, both synaptic potentials are the same amplitude, then the plasticity index equals 1. But if we have synaptic facilitation and the second one is bigger than the first one, then this index will be greater than 1. And if on the contrary we have synaptic depression, then and the second the amplitude of the second action synaptic potential is smaller than the first one, then this index is lower than 1. So here I'm going to show you this index of plasticity plotted against the interval between the two pulses. And here we have an example recording of where we gave two pulses separated by 200 milliseconds. As we can see, we got facilitation, and that's the plasticity index. And as we increase the interval between the presynaptic impulses, we get extinction of facilitation, which is nothing new. It fits to a single exponential. But when we gave two pulses with intervals higher than 800 milliseconds, like in this example is 1 second, then we get depression, and depression increases with the interval. So, what's this?
Small question: what happens if you take interspike intervals, which are of the order of what would happen inside. So very high frequency. So if you take interspike intervals of the order of 5 to 10 milliseconds,
You get facilitation.
it's facilitation.
Yeah.
Okay. Nothing
Yeah.
strange happening there.
No.
Okay.
No, it's just that to have clean action potentials, we give pulses of 10 milliseconds.
I see.
So you can't really. You can, but it's not.
It's clean.
Yeah. The bridge is not so well balanced, and yeah. But yes, there is still facilitation. Erin?
If you change the amplitude of the pulse you deliver, do you change the cutoff points for facilitation, or does it stay the same because of the all-nothing
They're all,
nature of the
all
event?
of nothing.
So
Yeah.
you can increase the pulse delivery to the presynaptic cell, and you'll still get the cutoff at 800.
Right.
Okay.
Oh, I mean, we didn't even do that, but action potentials are all or nothing. So,
it does-
we just ensure that we get above threshold to get an action potential
So the
with
cell doesn't care
each
how
pulse.
it fires, it just cares that it fires, yes or no.
Yes. Yeah, and the amplitudes of the action potentials are the same once you pass a threshold.
Have you changed the calcium concentration in the preparation
Yeah.
down? And what happens to the
No, not
to the
down.
lung stimulation?
No.
Nothing.
No, we didn't. We
Oh, you
didn't lower it.
Ah.
We increased it.
It may be just the example you're showing, but it looks like there's a leakage in not in the current, not in your electrodes, but in the synapse.
What do you mean a leakage?
This postsynaptic potential doesn't look like a unitary potential. It looks like there's a couple of unitary
yeah.
potentials along the way.
Yeah.
And
Yeah, we always see that, and that's something else we would like to study. I think that's caused by two synaptic entries on different positions of the same branch, which arrive with a delay to the soma. But yeah,
The other possibility would be that the same synapse that you're stimulating, or the same synaptic contact that you're stimulating, still produces. So the calcium that is in the presynaptic terminal is still producing releases that are not that big in the second that you're waiting for the next potential. And maybe that residual calcium is doing something to the calcium machinery so that you see the depression later on, but it's because of a saturation effect or something like that in the presynaptic terminal. Maybe the
calcium
release of-
saturation. Yeah, it could be. I mean, but in several preparations, it has been shown that two presynaptic pulses produce the same current,
Yeah.
calcium current. So I haven't done it in this precise preparation, but I'm just assuming that it's the same. But yes,
And so what's the
Things that
cutoff?
can happen.
Like, so there you show 200 milliseconds for the difference in the presynaptic pulses, and
This-
then you waited for a second to see the depression. So where do you start seeing the depression? Like half a second or?
800 milliseconds.
Oh, sorry. Okay.
Yeah. And it recovers in about 1 minute.
So that's why I was asking about if you increase the current amplitude on the presynaptic side,
You make
can
it
you
go back.
shift the cutoff?
Mhm.
In other words, is it paying attention for some reason
To the
to
frequency.
how it fires and the strength of the stimulus? Or is
Mhm.
it just paying attention to that there was one and it fired?
So we can't change the amplitude of the synaptic, the action potentials, but we did, I'm going to show you several manipulations to increase the initial release. Okay, something else? Okay. Okay, so this is where biologists need the help from mathematicians, and and we used a model previously published by Hennig, which accounts for the use of synaptic vesicles, N, in the terminal, and the changes in the probability of release, which basically depend on calcium. And this is how N changes after one action potential. It decreases because vesicles are fused, and then it recovers with some time course, while the probability of release increases and then decreases exponentially. And with this model, we got the necessary parameters to find something like our system. And then we simulated what happens with different intervals between impulses. And the model basically very nicely reproduces the decay of synaptic facilitation, but it does not explain this depression that we see with longer intervals. So what's happening? We have depression after a pulse, and we know that if we give a second action potential here, we get facilitation, so the synapse shouldn't be depleted, the vesicles shouldn't be depleted. And to test for that, we produced a third action potential after this second one that produced depression, and we get facilitation. So
second go to this noise?
This?
Yes.
Yeah. Yeah, we have to amplify a lot to measure these events because they're very small. This is 3 millivolts, whereas this is 30, right? So yeah, noise is clearer there. So the presynaptic terminal is not depleted, right? We have enough vesicles to even produce facilitation. This facilitation is even higher than the first synaptic potential. So what's going on? This is just the mean of several experiments like this. And we did simulations with the model with this 3-pulse protocol, and in order to get this facilitation in the third pulse, the model predicts much less depression than we actually get. And in order to get, and if we get this depression, then the model doesn't produce so much facilitation. So this is not explained by these parameters that are taken into account in the model. So it seems that
if I write clearly, if you set the model to have depression after 1 second, you don't have the initial facilitation. So if you go was like that. Right? Okay. So if I use the model with these blue dots, I will never get depression.
never.
So to get depression, you don't use this parameter. You
Right.
use even
You have
other parameters.
right.
Okay. So the whole picture is completely lost in any case.
Yeah. Yeah. If we get the right parameters to get this facilitation, then we don't get as much depression. And if we get the parameters to get this depression, then we don't get this facilitation. So apparently, there is something different to what we already know. So we tried to manipulate these parameters in order to see what is going on. And there are ways to change these parameters. The probability of release, as I said, can be changed by changing the extracellular calcium concentration. So we increased it to 10 millimolar. That's a 5-fold increase, more or less. And to increase the use of the vesicle pool, which is something I think similar to what you suggested, Erin. We, instead of producing just one initial pulse, we produce two to increase the initial use of the vesicle pool. And then after 1,000 milliseconds, we produce a test impulse. So here I'm going to show you a plasticity index, as I showed you before, but this is calculated with respect to initial release. So in the cases where we gave two initial impulses, we summed the amplitudes of the two synaptic potentials that we gave initially. And this, I'm going to plot it against this initial release, which is again the sum of the two initial amplitudes. So this is what we already saw. We have depression with two pulses in normal calcium, and that's the depression. And if we increase the probability by increasing extracellular calcium, then the first amplitude, the amplitude of the first synaptic potential is bigger, and we get more depression. Remember that here depression grows downward, right? So
What is A2?
So here there is no A2, but in these cases A2 is the second action synaptic potential. So if we increase the use of the vesicle pool by giving two initial impulses, and now we sum these two as initial probability, then we get even more depression. So this tells us that the amount of depression does depend on the initial use of the vesicle pool, right? Nevertheless, the appearance or not of synaptic depression does not depend on how much the vesicle pool is used. There is synaptic depression no matter what. And if we manipulate both parameters by giving two initial impulses with high calcium, then the depression in this case doesn't increase anymore because the synapse is already as depleted as it can be. Okay. So what happens in the case where we give a third action potential after the one that was depressed? Now I'm showing you the dots that I already showed are compressed here, and these are the results for the third or fourth synaptic impulse. And here the facilitation that we get with the following impulse doesn't change very much with the previous use of the vesicular pool. So facilitation is basically depending just on what we already know, calcium. But depression does depend on the previous use of the vesicle pool. So we have apparently two different causes for this depression. Although the amount of depression does depend on the amount of vesicles that were used previously, the fact that depression occurs just depends on time. So we still don't know exactly what the mechanism is, but we are proposing a model where we have a readily releasable pool of vesicles and a reserve pool. And when we give one action potential, some of the readily releasable vesicles fuse and release. And this action potential prepares some more vesicles, probably from the reserve pool or probably from the same readily releasable pool, to fuse. But and that if a second action potential arrives right away, then it it finds the terminal facilitated because it has more vesicles available, and there is an increase in probability because of residual calcium. But after some time, if another action potential doesn't arrive right away, after some time this is reversed somehow, and then a second action potential that arrives a lot of time after that finds less vesicles available at the membrane. And so that's what we can propose with our data. There was a very nice paper in PLOS Biology about a year ago that showed a depression mechanism whereby something that is released by the presynaptic terminal autocrinally activates presynaptic receptors and triggers an intracellular pathway involving phospholipase C. And this pathway eventually phosphorylates myosin light chain. And so myosin gets contracted and pulls vesicles away from the presynaptic membrane. So I think something like this could be happening, and we want to do experiments blocking phospholipase C to see if that explains this. But we also want to make a model that accounts for this. So I just want to acknowledge the student that did most of this work, Michelle, and Montserrat has participated a lot in the thinking of this and the funding agencies. And thank you all for your attention.
Are there questions?
Can you draw a schematic of what you just described so that I see mechanisms and
Sure.
I don't know, without any motivation?
Yeah, so apparently there's something, it's, I don't know, glutamatergic or whatever synapse, but apparently it also releases something else. Or maybe it's the postsynaptic which releases back or whatever, but there's something else released from somewhere that activates a presynaptic receptor, and this is coupled to a G protein, which in turn activates, it's actually in the membrane, an enzyme called phospholipase C, which takes a lipid from the membrane called inositol. PIP2. Difosfato de fosfatidilinositol. So this enzyme breaks this lipid and produces IP3 and the acylglycerol. And this in turn, I don't know if it's a direct mechanism or if it's through, it's through PKC. So this activates a kinase, which in turn phosphorylates myosin, the light chain of myosin. It binds a phosphate or two, and that makes, so myosin is a filamentous molecule. I mean, it forms filaments, and it's the molecule that makes our muscles contract. One of them. So when this molecule is phosphorylated, these filaments kind of contract, shorten, and if they are linked to vesicles, then they pull the vesicles away from the membrane, and so less vesicles remain here, and if another action potential arrives, there are less vesicles available, so you get a smaller synaptic potential.
So you have a model.
Well, I haven't tested that in my system. This is something that was published in another synapse, but which could well explain. Since it depends on time, I think it can depend on second messenger cascades, right? It makes sense, I think. But we have to test it.
And you would also like to have like an extension of the Hennig model, like a very abstract.
Well, I don't know what mathematical parameters we would need to add to the model.
I imagine a third variable.
Right. So that's your job, guys. Yeah, we should do that.
Other questions?
So what kinds of presynaptic calcium channels do you have in the membrane? Just in the terminal.
That's tricky in leach because
We'll speculate.
so usually in presynaptic terminals you have N, PQ types of channels. We have seen in another neuron that is a serotonergic neuron that forms synapses, that there are also, so in those presynaptic terminals there is the active zone with small clear vesicles, but these are surrounded by dense core vesicles, which we believe release in the periphery of the synapse. And our data have shown that in the active zone there are some channels, let's say NPQ, but it's tricky because these channels in the leach don't necessarily bind the drugs that you use to block these channels in mammals. So these are not known, but we know that there are L-type calcium channels, we think, here. And those are necessary to release the dense core vesicles.
Okay.
And also, these other neurons release also from the soma, from the cell body. And there, it's purely L-type channels.
I think you were going along the same lines I was thinking, whether some of this has to do with dynamics of channel inactivation or recovery
Exactly.
from inactivation, right? Which would make, if you have L types there, maybe. NPQ is not so much.
It depends. So what we were thinking is that apparently what we thought when you showed the data is that the first pulse starts, whether or not it's a second messenger triggered mechanism or something simpler than that. And an activation of the calcium machinery and the calcium channel population that allows you to trigger the activation and release. But then your experiments where you have the pulses after you wait sort of contradict that.
No, but it looks like
Yeah.
a recovery from inactivation. So you have your first pulse, which triggers
of.
an
That's why
activation.
it's
Your
sort of.
second pulse,
Which recovers.
Yeah.
which allows it
Resets.
to recover, but not yet. So you see a smaller potential. Then you immediately fire another action potential, and that
Exactly.
recovered population rebounds. So it looks like a recovery from an activation.
But it-
Yeah,
but
well,
it's
in
sort of
these other
contradicting.
cells, the serotonergic cells I was talking about, John Nichols showed that the paired pulses trigger the same calcium current. So,
There's no definition.
But if you have,
I mean, it's not the same cell, but
those are whole cell currents, right?
Yeah, the Nichols
Yeah.
currents are pulsar currents.
yeah.
What if you have microdomains?
Could be, yeah.
If you have microdomains of inactivated channels,
They are whole cell currents, but they were done in synapses in culture, in isopotential cells. So, yeah.
And there's
Yeah, I
no
don't
way
know.
to measure.
The calcium current in the terminals.
To do some kind of voltammetry in the synapse, right?
or what if you,
To see.
Voltammetry? For
Yeah,
what?
like you can measure dopamine. Why couldn't you measure whatever your? Is this cholinergic? What is it? What is the synapse?
Glutamatergic.
It's a glutamatergic synapse. To measure the amounts of glutamate that you see in the terminal, because if you see that the glutamate release is in line with the postsynaptic currents, then you can still you can trust that you should be looking presynaptically, but not otherwise. Right?
do the classic L channel blockers work in the system? Nifedipine,
For L, yes.
nimodipine?
Yeah, nimodipine.
Maybe you could put an L channel, I don't know, put an L channel blocker on there and see what happens. 'Cause I
On
wouldn't
this-
think if there's
on
any
this synapse?
inactivation playing a role, it wouldn't be the NPQs, it would be the Ls, right? So.
The problem is that this is an invertebrate, so it's, remember with Manduka it's the same thing with bees. So maybe you want to look into in the bee literature,
No, but why? Why are you?
because agonists have.
No, but
No, but nimodipine works.
That's why I said
Oh, okay.
nimodipine works.
Yeah.
So.
Well, that's a start.
We don't know how to block this, but
But I
Yeah.
don't think you
But-
need to. If you
but-
put the L type on, and if you put the L type blocker on and you see no change in this kind of rebound phenomena, then it's not the recovery from an activation, right? You could
From that type of channel.
from that type of channel, yeah. But that's the prime candidate. Your NPQs are not going to be doing.
I like the myosin hypothesis.
yeah, me too.
It's pretty cool. But I mean, just to rule out another possible channel, to rule out the possibility of channel dynamics. I like the myosin.
But that, yeah,
It's
we
much
can
cooler.
do that.
So if you're adding a third variable, let's say, it should be a variable that is both really slow in reacting to the presynaptic potential, so that the chain of events triggered by the G protein coupled receptor acts, and at the same time, it should be fast enough so that it doesn't matter when you get a second pulse right away. Because otherwise you get in trouble. You can't get both.
Well, I was thinking that you have a very fast variable and a very slow one, and if you have two pulses, the fast one will dominate, and the slow one will always be active, not sufficiently low. I'm just kidding, but yeah.
But it has to relax at some point.
Yes.
So
So maybe it's both. It's a G protein coupled mechanism plus the channel kinetics. The channel kinetics is the fast and the G proteins are the slow. I don't know.
Yeah.
don't know if you said it before, but why did you choose the leech as the biological model?
Because it has. I wouldn't say it's cheap.
Can't be the official version.
It's easy. It has big cells, few cells. The cells are always located in the same places in the ganglia, so you can visually identify them.
Easy anatomy.
Right.
Do you have any genetic tools in the leech yet, or not yet?
So it's big. It's starting, yeah, but it's much slower than mouse. Yeah. There's now a transcriptome and morpholinos work, but there are some tools, but not as much as in rodents. Yeah, or Drosophila.
Yeah, but Drosophila is tiny. That's the Drosophila. They have the same identified cells, and we have the genetic tools, but it's tiny. It's really hard to record.
Right.
So you don't have that advantage.
Mm-hmm. Yeah, these are huge.
How many hours do they survive once patched? Or once
A good recording can be held for 1 hour or so.
Okay, not that much. It's not like in
It's
the...
quite a lot.
It's, yeah.
Sorry, your face. But in the STG it's weak, right?
What?
In the STG they managed to record for weeks in a row. Am I wrong?
Wait, what's
I don't think so. Once impaled? I don't think so.
Okay, then...
No, because on top of that, you're not oxygenating, so it's gonna die. It's gonna die in a certain amount of time.
I mean, the ganglion with oxygenated water and sugar, that remains awake for weeks. I'm sure.
Oh yeah, we can culture the cells for weeks, yeah.
And then, okay, then it's extracellular recording.
But if you're asking me how long a recording lasts, an hour is very good.
Okay, sorry.
Yeah.
Like no rights.
You're sticking an electrode into the cell.
Okay.
The cell doesn't like that very much.
So maybe you know this literature, but there's quite a bit of recordings in the thalamus, and it's specifically thalamocortical synapses.
Uh-huh.
If I am correct, it's even better. It's ventrolateral thalamus to cortex. And there you see a phenomenon that is very similar to this one. But instead of seeing facilitation depression, you see the opposite. You just change it.
Mm-hmm.
Just flip everything around, and you see the same thing. But it gets better because if you actually do pulses, you do, for example, a pair, you wait, and another pair, and you wait, and you do this 5 times. The 6th time, so it's a long time. All of a sudden you see a response that is like this, and it's almost suprathreshold. It's almost like you get one synapse to make you
But
fire
that's
the whole
LTP.
neuron. And it's really weird because in the beginning people thought it was just an artifact. You're breaking your patch or something. And it turns out that what happens is that there's this superpotentiation of the synapse. In this case it's the opposite. It looks like, right? But it seems like it's a very similar phenomenon. If you want, I can give you some simulations with that,
Okay.
just with the opposite parameters, because what you need in this case, if you wanted to get that phenomenon, is to actually lower the, if your experiments, if what you think is happening to the probability because of the increasing calcium is happening, meaning that you're changing the probability with your increasing calcium, and the variable that is responsible to this is the number of vesicles in the readily releasable pool,
Okay.
then what happens in that case is that the steady state of the readily releasable pool is actually going up. In this case, it's going down. And it's recovering. The readily releasable pool is a variable that is recovering. If that is not what's happening, because there might be a couple of controls to do, right? Then you have to go on to the probability of release and not on the number of vesicles in the readily releasable pool, right? But there's a model already that does that kind of a thing.
Okay.
Your time course was a lot longer, wasn't it?
Yeah, but it doesn't matter because it's it's like 2 orders of magnitude longer, which is pretty much, Citlali is looking at hundreds of milliseconds, right? 700 milliseconds or so. And so what's happening is that in this case, in fact, you take a model that is very similar to the Henning model, super similar. And what you do is you lower, in your case, I don't know what they called it, n infinity. They called it 1, actually. They have it at 1. They have a steady state for n at 1. That's the Henning model.
Yeah.
So instead of 1, you call it f, and f goes down with the first pulse. And it takes quite a bit to recover towards 1. And when it happens, you see this thing with the second pulse, but not otherwise. And in the local dynamics of n, what happens is that you still release in the first one. You can't release the second time. But if you wait long enough, instead of releasing, what happens is that the releasable pool has
Increase.
disappeared.
Okay.
Which is what you're describing. It's just
Yeah,
that you're not
in my
putting
case,
the
yeah.
explicit. It's not a Mayos independent thing. It's a whatever the fuck it is, dependent thing. And we're in the internet. So something like that. And so there's already a description that might fit the bill for this kind of thing. And the problem is that you might not have a way to justify the molecular mechanism underlying this thing unless it is something similar to what these people publish. Because we don't know.
Yeah.
Right? And so that is the reason why we never published that one.
You did?
Because we didn't have a way to justify the increase in the steady state number of vesicles in the readily reducible pool we couldn't justify the increase in this case. That is sudden accumulating but sudden in the readily reducible pool.
Yeah.
In your case it's the opposite.
Yeah, but if you see that in such a long time, I mean much longer than this, couldn't that be due to transcription, gene transcription, and
The time of the experiment, so for the 5th pulse, 6th pulse, when you start seeing it, it's no more than 3 minutes.
No,
okay,
then it's sure.
yeah.
Yeah, that was one
But
of the
she's right that
observations.
there's other mechanisms that could be jumping in.
Yeah, because the PhD student, maybe you knew him, Mario. He was doing experiments that were much longer than that, and he was arguing that if you waited long enough, he would stop seeing crappy stuff like this, right? And so the first comment was, well, you're allowing gene expression or regulation, transcription regulation, or something that happens that maybe explaining it. So he went back, he redid the whole data, and he still saw this thing. And he didn't publish it, and later on somebody published it, and he was beating himself in the head because it was
Yeah.
it's like you have activation that is more or less constant on a synapse, more or less periodic, and you make that synapse be the sole responsible for the action potential production in the whole cell. Because you generate a huge pulse after a little bit. Something like that. In this case, it's the opposite.
Yeah.
But it's the same phenomenon, I think. Dynamically, it's the same phenomenon. You're moving where the steady state of the number of readily reducible vesicles is. In this case, you're moving it down. And then it recovers slowly. And when it recovers, you release again the number of vesicles you should be releasing. Right? That's what you're describing, if I understand correctly.
I mean, there's a decrease in the readily releasable pool just because of the action potential, because some vesicles are fused, are used. So that's a decrease. But that doesn't account for this depression. And there's something else that activates with long times.
Yeah.
Do you have a PKC blocker you can drop in there?
I have a PLC blocker. Yeah,
That would be a cool experiment.
Yeah.
yeah,
For this
I know.
Because the only other way, and that's super difficult, is like a freeze fracture EM, right? So you freeze it at the point, take the M, and then you can see where the docking of the vet, but that's...
Do you know anybody
Yeah.
who's with freeze fracture?
No.
No? We don't.
Maybe Jack McMahon knows how to do that.
And you have
He's
a
an
good
expert.
relationship with him?
Yeah,
I know how to do the EM part, not the freeze fracture.
Are we still being filmed?
Maybe for later.
Thank you very much.
We can edit it, so it's not live. So if there are no more questions, let's thank again Svetlana.
Thank you.